Welcome back friends, welcome back in another video lecture from microbiology with Shagni. In this video lecture, I am going to talk about baculovirus expression vector. This video is simply the continuation of the previous video where we have talked about the basic description of baculovirus. Okay, so in this video we are going to talk about the basic mechanism of baculovirus expression vectors. Okay, so let's start it. The first thing is that if we carefully study the overall genome of baculovirus, then we find out that there are some genes such as polyhedrin gene and stuff that are not necessary for their tissue culture life cycle. Okay, so the main idea of baculovirus expression vector is that we are going to replace this non-necessary gene of baculovirus such as polyhedrin gene with our foreign gene. Okay, but the main challenge here is that baculovirus genome is too large to insert the foreign gene. So what is the solution of that problem? So we will instead use another transfer plasmid. Okay, so what is going on? Transfer plasmid is constructed with our gene of interest along with the region the derived from polyhedrin, along with the sequence derived from polyhedrin region of viral gene, so that it will be easy for homologous recombination that we are going to talk about. So transfer plasmid along with our gene of interest are and our baculovirus genome, suppose ACNPV. I have talked about what ACNPV in my previous video. If you do not know what is ACNPV, then I will recommend you to please watch that video and then come back here again. Okay. So the transfer plasmid and along with the ACNPV, that is our baculovirus genome, is co-transfected into a SF cell, that is a insect cell. Okay, or you can say these two things are co-transfected in a insect cell. And if you want to know about what the full form of SF is, it is quite difficult. So that it is Spodoptera frugivora. So it is simply a insect cell. Okay. So we are going to co-transfect our transferred plasmid, which consists of our gene of interest along with the Autographa californica nuclear polyhedrosis virus. This is the most extensively studied baculovirus strain. Okay, so after co-transfection, the foreign gene and the polyhedrin gene come close together and they undergo a homologous recombination. Here homologous recombination should be a double crossover recombination, so that our gene of interest will successfully knock in our baculovirus genome, and the unnecessary polyhedrin gene will successfully knock out. Okay, so what we have now? Now we have baculovirus genome along with our gene of interest. Okay, and as it is present in the infected cells nucleus, we can say infected cell nucleus, then the first job of baculovirus genome is to shut off the host gene expression and use, then they will going to use the host cells machinery to transcribe our recombinant DNA along with the translation of our recombinant DNA to give us the recombinant product. Okay, so what is going on? The host cell machinery help us the transcription of our recombinant DNA along with the rest of the baculovirus genome. Then the translation will occur in presence of ribosome, and then we will have our recombinant protein. Okay, this recombinant proteins is overexpressed. That is one of the most important feature of baculovirus expressing vector. That means the protein what you will going to have is disulfide bond containing, that means disulfide bond will be present in case of cysteine residues, plus it will be oligomerized That is the second thing. And the third one is, and the protein will be properly folded. That means the protein that we are going to have here is of properly in quaternary structured. Okay, that means it is well expressed. That, and not only that, it will another important feature of baculovirus expression vector is that it will have the ability to undergo some post translational modification, such as O-linked glycosylation, N-linked glycosylation, acylation, amidation, isoprenylation, all that stuff. Okay, so the distinguishing factor of baculovirus expression vector from other highly eukaryotic expression vector is that it will provide the high expression of clone gene rather than the other expression vectors of the other high eukaryotic expression vector that are present. Okay, so that is the overall idea of baculovirus expression vector. Here the transfer, the recombination event is going on. That is mainly a double crossover recombination event, and it is homologous event is going on between the foreign gene and polyhedron gene. This polyhedron gene is the unnecessary part of the baculovirus genome, which is knockout, and our foreign gene is knocked in. Then with the help of host cell machinery, it will translate it into the recombinant protein, that is our desired protein, which is well folded over, which is overexpressed. That means it is well folded, disulfide bond is also there, and oligomerized. That means a proper quaternary structured protein we will going to have, and it will also exhibit some post translational modifications too. So that is the overall idea of baculovirus expression vector. So I hope this video is very helpful to you, and if it is, please like the video, share with your friends, and subscribe to my channel. Thank you.